welcome back to my YouTube channel. This is Sowmiya, and as you guys know that I am a biotechnology student. As a student myself, I've gone to YouTube searching for topics that are to be understood by me, and seeing kind of video presentations has always helped me more in understanding the topic. So I thought, why not take videos for helping other students? So I'll be starting this lecture series with a subject called molecular cloning techniques, in which the unit 1 comprises of cloning vectors. So I guess we can go into the video. And to start about cloning vectors, you need to know what is molecular cloning first. So molecular cloning is a set of methods or techniques which are used to insert recombinant DNA into a vector. So before going into the topic, you need to understand what is a recombinant DNA, what is a vector, and you'll be seeing all of that in today's lecture. So as a definition, you can say the process where cloning and manipulation of genes requires the ability to cut, modify and join genetic material and also check the parameters of the molecules. So the process where you will be cutting, modifying and joining genetic materials, and then you clone it, and then you see the expression of the gene that is inserted. So this process is known as molecular cloning. It can be also called as genetic engineering, genetic modification or gene manipulation. So now we've understood what is molecular cloning. So now we need to understand what is the purpose of this cloning method. Why do we need to clone? So we have two major reasons why we need cloning. So the first one is the purification of gene of interest. So you have a gene that is need to be purified, means you are isolating the gene, you are inserting into a vector, and then you are sending the vector into a host cell, and replication happens and clones are produced over the time with the gene of interest. So your particular gene of interest is purified in the clones. And the second major reason is manipulation of protein synthesis. When we need to produce protein in a large scale, or when we need to influence or manipulate biological pathways, and when we need to change the protein structure and function, we go in for gene cloning. So basically, you guys know the central dogma from DNA to RNA, and RNA is then expressed as proteins. So when you change DNA, subsequently after the transcription process, the RNA changes, and after the translation, the protein. So hence you can manipulate the protein synthesis with your gene of interest. So as I've explained what is genetic engineering or molecular cloning and what is the purpose of gene cloning, now we are going to see the steps involved in gene cloning. So the basic steps involved in gene cloning is that you need a vector. So talking about vector, you need to know what is a vector. So vector is nothing but a self-replicating DNA molecule. So this vector is nothing but vector is also a DNA molecule, but which is self-replicating in nature. It carries DNA molecules that needs to be transferred to the host cell and can be purified through clones. So a proper definition of a vector can be given as an autonomously replicating DNA sequences can be used to carry foreign DNA fragments. So that is all about vector. And why do we need a vector, and the features and the structure of a vector, we'll be seeing it in the next video. So first you need a vector, a circular autonomously replicating vector, and then you need the fragment of the DNA to be cloned. So the fragment of the DNA to be cloned is the gene of your interest, and now you cut the vector, you place the fragment of DNA to be cloned, and then you ligate it, we get the recombinant DNA molecule. And we'll be seeing all the enzymes ligation process in the upcoming videos. So for now, what you need to know is you have a vector and you have the fragment of DNA that is your gene of interest, and then you are inserting it, and now you get the recombinant DNA molecule. So after you get the recombinant DNA molecule, You are taking a bacterium, which is the host cell where the replication of the rDNA is going to take place. So after inserting the DNA molecule, this process is called transformation, where the bacterium takes up a foreign DNA molecule, that is the vector, with our gene of interest. So the entire vector is a foreign DNA molecule to the bacterium, and this process is called transformation, where a bacterium takes up a foreign DNA. And now this bacteria is carrying the rDNA, that is the vector with the gene of interest. And now the multiplication of rDNA takes place inside the bacteria, and now the division of host cell takes place, which is the common replication process in any host. And now we'll be plating the bacteria carrying the rDNA in a solid medium, because as you guys know, bacteria also requires a nutrient medium to grow, and for its replication process we'll be placing the host cell in a solid medium to produce n number of clones, and we can purify our gene of interest from there. So this basically is the entire process of gene cloning. So till now we have seen what is gene cloning, what is the purpose, and now we've seen the entire steps in gene cloning. So as I've said, we're going to be seeing a very short introduction to the tools of gene cloning. So as I've said in the definition before, the tools are required for gene cloning are basically enzymes. We know what is molecular cloning. It is a technique where we are able to cut, modify, and join genetic materials. As I've said, in a vector, you have to use enzymes for cutting and then joining our gene of interest. So we'll be seeing what are all the enzymes and how do we choose an enzyme for this cloning technique. So the choice of the enzyme depends upon how easy, that is cost effective, it is to purify the enzymes, and the abundance of enzyme in the cell, and how easy it is to separate from other undesirable products. So we during the molecular cloning techniques, it has to be cost effective for us as well. So the cost effective is one of the major point, and the second one is the abundance or the expression of the enzyme in a cell. If the enzyme that we are looking for in a cell is produced in very low quantities, we have to go for other viable options, because we cannot go for that enzyme which would be not cost effective for us. And the third one is how easy it is to separate from other undesirable products. This is basically a downstream processing where we'll be separating our desired enzyme from a mixture of all other enzymes which we do not require. And the second point is how well does it do the job. The main job of the enzyme is to cut and modify and join genetic material as said above. So how specific or accurate the enzyme is, because the enzyme, I'll be talking about the structure of the vector in the next video, but the enzyme cuts specifically in a place in a vector, and that is where our gene of interest is joined and then ligated. If the cutting does not happen in a specific place, then the replication is not possible. So we have to see how specific or accurate the enzyme is, and then we'll be seeing about the specific activity or the speed of the enzyme, and we'll be also looking into the details of the reaction which it catalyzes. So that is all about the choice of the enzyme. Now, the enzymes can be classified based on its activity into two different types. The first one is exonucleolytic activity. So we all know the term exo means outside. So it can be defined as the dismantling of the molecule bit by bit from the ends. So exonucleolytic activity is where the enzyme would be acting on the ends of the molecule. And the second category is endonucleolytic activity, which is defined as cutting within the molecule. So I think it's very clear. All you need to remember is exo is from the ends and endo is within the molecule. That is, it starts cutting inside the molecule and not from the ends. So the widely used enzymes in cloning techniques are restriction endonucleases And endonucleases, I can say that as cutting within the molecule. So we'll be using enzymes which starts cutting within the molecule. And these restriction endonucleases are a part of the natural defense mechanism against incoming DNA in bacteria. So, talking about the natural defense mechanism against incoming DNA, bacteria, no, not only in bacteria but also in humans, when a foreign molecule or a foreign particle enters our body, it is a natural, our body starts exhibiting natural defense mechanism to push out that foreign DNA molecule outside our body, because any foreign molecule may cause harm to our body. So it is also natural in prokaryotes to, exclude the incoming DNA from their body. So the bacteria contains restriction endonucleases. So that is where we purify our restriction enzymes from bacteria, because bacteria contain restriction endonucleases which are part of their natural defense mechanism. So we purify and extract this enzyme from bacteria and use it for molecular cloning techniques. Restriction enzymes can cleave viruses as well as plasmids. So viruses, they cause harm to bacteria by invading the host cell, and they use the host cell for their own replication. Plasmids are the extra chromosomal material which we use to insert our gene of interest and to replication it, and then for purification of our gene of interest. Bacteria also have this special mechanism called methylation of DNA. Since they contain a natural defense mechanism by having restriction endonucleases in them, see, the main point of restriction endonuclease is to, degrade the incoming DNA. Bacteria also contain DNA. So how will the restriction enzyme differentiate between the host cell DNA and the incoming foreign DNA? So this is where a special process called methylation of DNA takes place. Methylation is nothing but the DNA in the bacteria is methylized. That is, a methyl group is added to the DNA of the host cell. So this methylation of DNA protects the host cell from degrading their own genetic material. So restriction enzymes does not act upon DNA which is methylized, and hence it cleaves only the foreign DNA which is incoming in the bacteria. So this is one of the main process where bacteria protects their own cell DNA by preventing degradation through methylation of DNA. So we'll be stopping here with this. So that is all the first part of cloning vectors include, and in the next video we'll be seeing why do we need a vector, what is a vector, a brief introduction to what is a vector, and the classes of vectors, and we'll be seeing the features of vector, the structure of vector, and how do we choose a vector for molecular cloning techniques. So I hope you guys liked this and understood the concept of molecular cloning, the process of it, and how do we choose the enzymes, and how does bacteria protects its own DNA from degradation. And do let me know what kind of topics you would like me to take in the further upcoming videos. Even if it is of 11th 12th, I'll be more than happy to help. And do let me know in the comments on which aspects I need to improve. And if you guys need this PPT where I've prepared, I'll be mentioning my phone number in the description. Do let me know and I'll let you know how you can get this. This material is I have prepared on my own from the classes I have attended at my college, and I hope this video helps you guys a lot. Thank you. I'll be seeing you guys in the next video.